Okay. Well, welcome everyone to the Funga Seminar Series. I'm Colin Averill. I'm the founder CEO of Funga. We restore fungal biodiversity to forests to make them grow more and remove carbon from the atmosphere. It is my pleasure today to introduce Dr. Kitty Gehring, who's professor of biological sciences at Northern Arizona University, where she also co-directs the Center for Adaptable Western Landscapes. She's been recognized as a fellow of the Ecological Society of America and has also received the Francis B. McAllister Award for Conservation and Education. She's authored over 100 scientific papers in her career, and her trainees have had a huge influence on our field, including Funga's very own Dr. Dawson Fairbanks. So, what draws a lot of our team to Dr. Gehring's work is her focus on understanding how the presence and composition of mycorrhizal fungi ultimately shape tree and forest function. In particular, her team has done really incredible work, in my opinion, integrating mycorrhizal fungi and soil communities into the practice of Western forest restoration, often with a focus on drought tolerance and in overcoming invasive plant legacies. Her work of using whole soil communities is especially of interest to the team at Funga. At Funga, we really emphasize these whole microbial community transplants in our own work and practice in the southern US. And so, for these reasons, and honestly, a ton more, we're super stoked to have her here. So, please join me in welcoming Dr. Gehring to the Funga Seminar Series. Yeah, we got some emojis, emoji hands. Perfect.
All right. Thank you. Thank you for that great introduction, and thank you for the invitation. I really appreciate it. And yeah, I'm going to sort of take you to some very dry places, which is where we work. And so I'm interested in your perspectives because we have potentially some different challenges than the ones that you all are working with. So I'm sharing my screen. I want to make sure that everybody can see it okay. Good. Okay, so given your interests and some of our recent work where I really sort of think about it as trying to put mycorrhizae to work, given that we know so much about what they can do, we want to see if we can get them to actually do that, some of those in some of the worst places to solve some of our terrible problems in the West. And some of those problems that we're facing are, we're in the southwestern United States is in the midst of a mega drought. And that's been going on since the early 2000s. So we are incredibly dry. And that we also have problems with invasive species that carpet the landscape, like these in the sort of, I guess I can use my right here. Oops, excuse me. I don't know if you can see my pointer or not. Yep. Okay, good. I haven't done this Zoom teaching for a while. So we get carpets of non-native grasses that then also are really important in fueling fire. So we have a really big problem in the Western United States of increased frequency and intensity of wildfire, and they don't just burn a few acres, they burn hundreds of thousands of acres. And then that leaves us with really sort of landscapes that are very different than they were, and very difficult for plants to reestablish, especially the native communities, and especially when you have these non-native grasses that really sort of set up this grass fire cycle where they are good at growing quickly in sort of nutrient pulses, die off early, and then have these fine fuels for more fire. So we have sort of some big challenges, and what we're investigating a lot are sort of basic questions about mycorrhizal fungi and resilience for ecosystems, sort of looking at how they change with these stresses, and then also trying to see if we can use mycorrhizal fungi and associated microbial communities in restoration. And I'm going to give you just sort of two examples. One is a study system, the pinyon juniper woodlands, and the pinyon pine is ectomycorrhizal. And so this is a picture of its cone here, and some ectomycorrhizas. And it is often the only ectomycorrhizal host in those landscapes. And the rest of the plants are arbuscular mycorrhizae. mycorrhizal like its co-dominant, which in this case is Juniperus osteosperma. That's the one I'll be talking about. There are several pinyon and several juniper species, but I'll be talking about Pinus edulis and Juniperus osteosperma in the example here. So we'll be looking at both of those groups of fungi. And the second example I'll use is with Populus fremontii, which is Fremont cottonwood, and that is a dual host. And so we can see that it has arbuscular and ectomycorrhizal fungi at the same time in the same plant. And my first example will focus on pinyon juniper woodlands in Colorado, and specifically near within Mesa Verde National Park, and also surrounding lands in that area. And again, it's pinyon juniper woodland, and Mesa Verde National Park is famous for its cultural resources, which you can see these images on the from the park brochures here. Um, but it's also well known for its sort of old growth pinyon juniper woodland, which is not, there are many, many old trees. And when you look historically, there's no evidence that it was an environment that burned regularly. So there's no really good, evidence of fires being a really big, important part of these pinyon juniper woodlands. And so when you have fire, there isn't really a good strategy by that existing vegetation to return. And so here's a picture of one of those stand replacing fires, and they now cover over 60% of the Mesa Verde land area. So this project is really Mesa Verde, Ute Mountain, Ute lands around there, and then it's in cooperation with Sasha Reed and John Bradford and their groups at the USGS as well. And this is my student, Jillian Trimber, this is her PhD project. And the first thing that we did as a team was to just look at 3 of the fires. So here's a map of the fires that happened all in the early 2000s. Some of them have reburned. There's also new fires, but again, fire has impacted in terms of killing all the trees, about more than 50% of the land area of the park. And then, of course, it extended beyond that too. And then just looking 20 years later, do we have any trees returning? And what we see is that we didn't find a single tree seedling excuse me, in any of these 3 fires that we surveyed, even though we would see tree seedlings in companion unburned areas. So it wasn't like there was no seeds produced. It was that the conditions were not good for those to be establishing in the burned area. And of course, our minds went immediately to below ground factors. And so we looked at both the fire, and then we also looked at what the landscape became, which was mostly grassland and shrubland. And the grasses were mostly cheatgrass, Bromus tectorum. So this is one of these non-native annual grasses that establishes well post-fire. And then the second grass was western wheatgrass, which is native but uncommon at Mesa Verde, and it was planted on purpose by the National Park Service in order to reduce erosion, because on a lot of these slopes, what happens is fires happen in June. Then we often have a monsoon season, so a rainy season beginning in July. And if you don't plant something that grows quickly, you will lose a lot of soil off the slopes. And it's an incredible amount of soil loss you can have. So they had good reasons to plant this, but it has established very well along with cheatgrass. And so we wanted to know how much of it is the burning and how much of it is the grasses that have come in that are retarding sort of regeneration by the trees. And we expected that the fire and this post-fire vegetation would have some effects on arbuscular mycorrhizal fungi, but all of these plants are hosts for arbuscular mycorrhizal fungi, so we expected it might not make as much of a difference for juniper as it would for pinyon, because none of these plants are hosts for ectomycorrhizal fungi. So we expected that bigger effects for pinyon than juniper. The way we've approached this project is we've gone and sampled at in association with those three fires where we have the plant community. We also sampled biocrusts as well as arbuscular and ectomycorrhizal fungi, um, sort of to see what the changes were. Then we also did a couple of greenhouse experiments, and those focused on pinyons. And then we did a restoration trial using both pinyon and juniper. And I'm going to focus here really on the greenhouse experiments, where we're trying to understand both the legacy effect of the grasses as well as how they interact with pines because they're still there. So plant-plant interactions, and then I'll talk about the restoration trial. And so here's the first greenhouse experiment. And I'll just talk about one of the ones that we've done. And what we did was take one of these fires and just to, so you can see the image here of the unburned pinyon juniper woodland and the burned pinyon juniper woodland. And we took soils from each of those places. And here's what they look like. And you can see this really stark line. And we've actually looked for seedlings thinking they might sort of come out, that you might see them right along the line, right along the edge, because there might be seed limitations, but it's just this very stark line where it burned. And so we don't see any regeneration at the edges either. Okay. What we did for this experiment was basically take these soils, burned and unburned, and then grow pinyon seedlings with different neighbor treatments. So either another pinyon, um, cheatgrass, which is in yellow, and western wheatgrass, which is in blue. And then we took a look at both their growth, which I won't emphasize, but also their fungal communities so that we can see how different they are and if the grasses change them more than just the fire itself. And so we'll start here with pinyons growing with pinyons and take a look at their fungal communities. And what you can see is we don't have a very diverse fungal community. We have mostly two genera. One is Geopora, which is shown here. This is one of its sporocarps. We have several members of that genus. And then the other common one that's in the red colors is Rhizopogon, which is a hypogeous fungus, which is known to respond to wildfire. So it's known to be an important part of pinyon juniper woodlands and to be responsive to fire. So it's not surprising that you see more of it in the burned areas than the unburned areas when you have pinyons growing together. And now let's look at the grass effect. And what you can see is you get much more dramatic shift in the communities when you add the grass neighbor into the situation. And so you end up with almost all Rhizopogon in the burn soil that colonizes the pinyons, and much more sort of Geopora in the unburned soils, but still very negatively sort of affected in terms of Geopora abundance when you end up with the grass neighbors. So we think it's an interaction so that even 20 years after the fire, there's still an effect of the burning, but that a big part of that might be the fact that you have these grasses. And this is important to the seedlings because if you look at how much Geopora you have, which is on this x-axis here, and how well you grow, the more Geopora you have, the bigger you get. So this suggests to us, and we see this in others of our studies, that Geopora is really important for drought tolerance as well. And it seems like for whatever reason, we don't know the mechanism, that it isn't doing, even though it's present in the burned soils, it really is not doing well at colonizing roots in the presence of these grasses. And one thing we did observe, which is in the paper, some pictures of it in the paper, is that when you see the ectomycorrhizas in these areas with high amounts of grass, you see that the root tips actually are just dead. So you see a lot of dead ectomycorrhizae. So it seems to be potentially sort of a toxic effect, some kind of chemical that the grasses might produce. And we know that we're doing some metabolomics on cheatgrass in another study. So we think it's producing some metabolites that might be harmful to ectomycorrhizae. So our next step was to start the restoration trial. And so could we do like things like you all do, which is to restore these missing fungal community members and then bring them back into the burn landscape. And would that help the plants at least survive there? We didn't look at establishment itself. So what we did is we don't want to take a lot of soil, but we took a small amount of live soil from the unburned areas, and then we grew plants in the greenhouse, not these particular ones. This is just a generic greenhouse shot. But we grew host plants in the greenhouse, so pinions, junipers, other AM hosts, other ecto hosts. And we basically, in that way, we grow them in these big tubs. We essentially, we call it bulking up the inoculum so that you're not using so much of the soil from the unburned areas, which are rare, and Mesa Verde National Park doesn't want us to sort of dig up the remaining old growth forest. So we take a small amount. We actually happen to use where they were making new parking lots and new trails. So we could just use soil from trees that were going to lose their lives anyway. Then we either add plants and grow them in the greenhouse for a while, or we directly apply the inoculum into the field when we're planting. And for this example, we actually grew the seedlings for 3 months in the nursery. And then just to remind you, so we did EM hosts for pinyon to bulk up that inoculum to get more ectos, and we did AM hosts for juniper because they need different things. So they had different inocula that they received. This is the design. So we know from other studies in these Western forests that pinions in particular need to establish under a nurse. And so we had shrubs that had established in this area. And so we had a nurse plant treatment. We also had an open treatment to compare. And we know that it's much hotter and drier in the post-burn landscape than it would have been in the forested landscape. But we also had sometimes nurse objects can be used. So a big rock might serve the same function as a shrub. And logs have been shown to do that in some forests as well. So we had all these logs around. So we implemented a log treatment as well. Our junipers are notorious for actually not liking to be propagated in the greenhouse. So we had lower germination success for them. And so we only had a couple of treatments. We just had the shrub treatment and versus open. And the pinions also got water at establishment during their first year. And a couple of additional times. So we had a high and low water treatment as well. And this just shows you sort of, again, it's in the Long Mesa Fire area in this restoration site. All right, so that's the design. This is a little bit of what it looked like. So here's a log, and we always had a live or a sterile. That's great. Next to one another, so they're in proximity so that they're experiencing the same sort of environment. And we know they don't grow very fast, so their roots aren't going to touch for a long time. And with the shrub, you can hardly see the seedlings, so they're very well shaded. And this is what the open treatment looks like. And we didn't really know much about the dependence of juniper on arbuscular mycorrhizae, but this just shows you after 3 months, this is the live inoculum and this is the sterile. So they were already significantly larger, which we then measured and accounted for that in our subsequent analyses. So that's what it looks like. Now I'm not able to, oh, there we go. Okay, here we go. Let's look at the survival data, and we'll start with pinyon. So this is our survival over 2 years. And you can see that okay. The live inoculum here, the sterile inoculum here, high water, low water, and then the nursing treatments, shrub, log, and open. And you can see the biggest thing that matters for survival is whether you have a nurse plant for pinyon. So that matters the most. You can see when you run the models for this, you do see that live inoculum has a small effect, and we can attribute that to the size differences I'll show you in a moment, and that the watering had a small effect. But the big thing for survival for pinyon is not the inoculum, but it's really having a nurse. And the logs, even though we were hopeful that they might be an easy way to replant rather than the shrubs, they're no different than the open. I don't know what is going on. There we go. If we look at the juniper, remember we only have two treatments. We have the inoculum and then the open or the shrub, and junipers could care less, which is really interesting about any of the things we did. They survived equally well in with sterile and live inoculum and in the open or with a nurse. So that was encouraging in terms of being able to plant junipers anywhere on that landscape. When we look at growth, we see a different effect. We see that the nurses, this is pinyon growth, so this is end of the first full year in the field and the second full year. And it doesn't work so well for pinyons or for junipers to just measure their height because they don't tend to grow from that apical meristem just and get tall, like I think loblolly might or ponderosa pine might. They don't sort of grow that way. So we actually look at their shoots and measure their shoots and how those grow. And you can see here that when you look at this first year of growth, that the nursing treatments don't matter very much, but that growth is really influenced by inoculation. So if you have a live inoculum, after the first year, you're about twice as big, and then those differences persist into the second year. And the nurse treatment doesn't matter for growth. It matters for survival. Okay. And you do get more variability in the open because we have fewer seedlings that have survived. Okay, that's pinyon. And then we see the same thing for juniper, which we were more surprised by because we figured there would still be arbuscular mycorrhizal fungal communities there. And again, if you look, the nursing doesn't matter, but this first year, small but significant differences, and then the second year, much bigger differences. So we do get a big effect of growth by providing that inoculum from the intact forest. And we get basically seedlings that survive and grow pretty well for these species of plants, okay, with those inoculations. So what are the implications for restoration? One is that we can expect sort of long-term effects of the fire, and that's going to be influenced also by what you plant after the fire or what vegetation comes in after these fires. And that's going to affect your ability to restore. But that if you bring in these microbes from the unburned areas, they could be a valuable tool along with the other things that we know about the biology of those plants. And we were expecting it to be important for the ectomycorrhizal fungi, but we also saw that it mattered for the arbuscular mycorrhizal fungi too. And I think that's partly because we really don't know much about the arbuscular mycorrhizal fungal communities of a lot of the arbuscular mycorrhizal trees in the Western United States. And we just don't have any data. And we'll hopefully be publishing some of that soon. All right, just going to launch into the second example, which is Fremont cottonwoods along the Little Colorado River. And these historically had nice willow, cottonwood, riparian areas along the Little Colorado River, which is a tributary of the Colorado. Beautiful landscapes, but it has all been taken over by non-native species. And partly that's because of changes in water dynamics. In terms of regeneration for cottonwoods, they have a very narrow window of when they're able to regenerate. And so you end up having to plant them, but we're trying to figure out how to plant them better and if having help from soil microbes might be a way to do that. And again, they're dually colonized by both ectomycorrhizae and arbuscular mycorrhizae. And the species of non-native that we were targeting in this project was tamarisk, which is in its own sort of unusual family. We call it tamarisk spa because in its non-native range, it actually has hybridized. So it's two species that were introduced and they've hybridized where they wouldn't have hybridized in their native range. So it's basically its own thing. We've not ever seen it make any kinds of mycorrhizas. We've looked at a lot of tamarisk roots and we see dark sapotite endophytes, but we do not see mycorrhizas in any of our studies. So it seems to be non-mycorrhizal, which means that then over time, when it's been there for decades and decades, is that how are you going to have mycorrhizae persist? And we've definitely seen that as part of the legacy that it leaves behind. So less mycorrhizae. It also lowers the water table, salinizes the soil. It brings up water from deep where it's saltier, and then it has this very salty foliage. Birds get stuck in it. So no one likes to walk through it. It's not nearly like you'd like it to be if you're sitting under a cottonwood tree along a stream in the Western US. It does not replace our native vegetation in that way. And then there's also evidence that there's other chemicals that it produces that are in the litter aside from the salt. So it's not a nice non-native plant. And so we took advantage of a spot where the water table seemed fairly high. And this was my former PhD student, Lisa Markovchik, who led this study. And we paired it with the greenhouse study, which I'll mention why we did that. And we used a large team of people to plant basically thousands of cottonwoods. It's turned out to be more challenging of a location than we thought. Once we had a weather station up there, we realized it got almost as cold as Flagstaff and almost as hot as Phoenix. If you looked at our max and min, so it was very challenging temperature environment. It was also in drought during our studies. The soil turned out to have high pH and then really sort of variable salinity. So one of the plants that grows there that usually wouldn't grow anywhere in the West is pickleweed because of those high salt concentrations. So very challenging place. But when you look in the distance, you can see cottonwoods. So they occur there still. It's just ended up being a challenging place apart from the reasons that we knew, which was tamarisk and drought. And so this is the American Conservation Experience, and they did tamarisk removal for us. And we had our experimental design was to have this tamarisk legacy soil where the ACE crew took out the tamarisk. We also had an area, and we don't know why it was free of tamarisk, but even when you look at historical photos, there's no tamarisk. And so we were able to then say we have tamarisk legacy and no tamarisk soil. And then in this experiment, we wanted to look at cottonwood genotypes. So we took 3 populations that were local, so in the climate area of the restoration site, and 3 populations that were not local. And they were taken from warmer places in the hopes that they could handle the high temperatures and drought well. And we also did something unusual in that we planted them in really long pots. They were about 1 meter tall. And then we had to dig these big holes. But then because cottonwoods need to get to the water table, we figured that they would get there faster if they had big, long root systems. But that meant we couldn't sample the root systems very easily either. So that's why we did the companion greenhouse experiment to see if what we did with inoculations were effective. We prepped things in sort of the same way. We took live soil from, and this time this is important where we got the soil. It was from healthy cottonwoods in areas without tamarisk. Okay, and again, we bulked it up as we'd done before, but the important thing is that they were from a 3 degrees C warmer site. So we're matching the inoculum to some of those populations, but not others. Okay, so we're just trying to see if that, what happens with warmer site inoculum. And in this case, we added it at planting instead of before we planted those cuttings. And it's cottonwood cuttings rather than seedlings. And the companion greenhouse experiment, I'm not going to present data on that itself, but it did show that the inoculum, when we added it, increased the mycorrhizal fungal AMF by twofold and EMF by fivefold. So we could see that the inoculum had the effect that we wanted. So let's see what happened. And this is our no soil inoculation treatment, sort of as a baseline. And so this, I'm only going to show you survival data here. Um, percent survival. And this is in the no tamarisk area, and this is in the tamarisk area. So this is our soil legacy, and you can see there's a big effect of that soil legacy. And our blue is the local population, and the green is the non-local population. And you can see that the locals are doing much better than the non-locals, and there's barely any survival at all in the non-inoculated soil if you come from the 3 degrees C warmer places. Okay, so, and this is our 2 year, sorry, spelled survival run. And this is what happens if you compare the inoculation to the non-inoculation. So this is with our non-local soil community. It doesn't really benefit the local genotypes. In fact, it's not significantly different here, but it is a growth depression here if you add that 3 degrees C warmer inoculation. But the big thing is that look what it does in the Tamarisk legacy soil. It really dramatically improves the survival of that non-local stock if you bring their inoculum with them. And this relates to sort of when you're thinking about restoration and you're choosing what you want to plant. And there are many projects on this. I just pulled this one recently, although I'm not sure how much it's still going because of funding cuts, but where people are trying to optimize thinking about climate change, which is what we were thinking about. Should we use stock from places that are already warmer because they'll do better with climate change in these warmer places as places get warmer. And what our study suggests, and there's others that show this as well, that that non-local stock might not do very well if it doesn't have its own home microbes with it. And that's what this study showed as well. And so for us, that has implications for thinking about where we take stock for planting cottonwoods, and if we're going to inoculate, where do we get inoculum? All right, and so sort of lessons that we've learned from these two studies are that soil microbes can help with dryland restoration, and that it can happen in sort of surprising places. So people will often think, oh, well, there should be plenty of inoculum after 20 years. But what we see is that, well, maybe there is inoculum, but maybe it's not the things that you need for successful establishment. And that also plant genetics, we've seen this over and over again in our studies, are important to consider when trying to move inoculum around and plants around. And with that, I'd like to thank you for your attention and then acknowledge other students and collaborators and funders of the research I talked about. And if there's time, I'll take questions.
Thank you so much, Dr. Gehring. This was awesome. Um, we would love to take people's questions. Please, you can raise a hand on Zoom or you can just drop it in the chat and I'll read it out. Um, I'm happy to kick us off though. So I think, you're showing really compelling results that, reintroducing these soil microbes can play a really important role in the restoration of these ecosystems. What has your experience been like sort of talking with practitioners about, adopting this more broadly, like beyond these sort of, academic field experiments?
Um, So we have enthusiasm from local managers, like for example, at Mesa Verde National Park. The folks that we worked with, they are very excited about our results and they show, because people actually going to national parks will go, where are all the trees? Why are the trees not coming back? Because they're right around all the cultural resources. So they just are going through this hot burned landscape. And so they often get shown our restoration trial, but so far it hasn't had the traction to do things in a bigger scale. And one of the things with the Mesa Verde project that we think might work better is that you don't need nurse plants for juniper. You do need the AMF. So there might be ways besides planting seedlings, because what they often want to do is just plant seeds. They're like, because we have vast, I mean, the areas of land that we're talking about are tremendous. And so they want to be able to use a helicopter and seed. And so there might be a way with the juniper to do it larger scale because they don't need the nurse plants and they just need the mycorrhizae. And so if we can get them there, that might be a way to get people to go, okay, this won't be that expensive, because usually it's the expense of implementing this at the large scale that prevents people from being as interested in it so far.
Will Gardner, please take the floor.
That was an awesome presentation. Thank you.
Oh, thank you.
Um, Super cool, like looking at what we do in the South, similar to what we do in the Southeast, but somewhere dry. But I have a totally unrelated question about the juniper. I've
Okay.
read in a couple places that sometimes juniper can also have ectomycorrhizal relationships. Did you see any examples of that? And could that potentially help explain why you saw like a big growth, like that big growth difference? Could there have been some sort of ectomycorrhizal relationship that it was forming?
So two sort of answers to that. One was that the inoculum that we used was really cultivated for AM. So, it would have, yeah. So I think the growth effect, it wouldn't have there might have had some ecto inoculum in there, but it would, because it comes from the same soil source, but it was really sort of designed to cultivate, and when we use that inoculum, it's filled with roots and, AM roots. And yeah, so that's one answer. And then the other answer is that I have looked and looked and looked at all kinds of junipers, including some in Austin where there was an idea that they would have ectos, and I have not seen any ectos on them. And we have sequenced them and we have done the microscopy. And yeah, so I think occasionally that might happen, but I haven't seen it in the junipers that I've looked at. They're all very convincingly the roots are kind of short and clubby, so I think people think that, but I have yet to see it.
Awesome. Thanks.
Mhm.
I love those questions. There's always a debate of does this ectoplant form arbuscular mycorrhizas or vice versa. Um, Anyone else? Otherwise, I'm going to start calling on people. I'm going to call on Dawson.
Yeah, I can't figure out how to raise my hand. Let's get into it. I actually also had a question about the AMF fungal inoculation, and so that was a surprising and cool result to see. And again, thank you for the presentation. It's really cool to see everything laid out that way. But for the AM, how did you actually, you said that you kind of cultivated a arbuscular mycorrhizae, or like how was that done in the greenhouse?
It's
And
just
then
by using AM host plants. So it's just growing, because you can't just make AM and not, well, you can make it out of, but it really needs an obligate biotrope. So it really needs a host plant. And so we're just growing host plants so we kind of take these big bins and then we have a little bit of the field soil and we make sort of a parfait of sort of sand or turf-face or something like that. And then we layer on the live soil, and then we let things grow in there for quite a while. We put it at high light, decent water to create the conditions for good exchange. And then we get lots and lots of roots and hyphae in the soil. And then we use that. And then like in the cottonwood example, we would just take little measuring cups and we had high school students. So we had actually prison crews dig the holes, and then we had high school students come fill them with plants and dump the live or sterile inoculum in from those bins. So that's how we were doing it with just small amounts of inoculum.
okay.
I
And then
saw
kind of
pictures of your operation with the heavy machinery and stuff, which is, yeah, very different from are kind of what we've done.
Yeah, I think one of the questions we have about like if we can inoculate with AM, so I'm always interested in that. And then I guess kind of more of a technical question. I was just curious about some of the primers that you use for getting both AM and EM in this study, or like the sequence.
Um, so I wish, I keep hoping that someone will figure out the primer, the one primer that does it all, right? But we just end up using the SSU primers, the for the AM, and then we use ITS 1F and 4 if we're doing Sanger, and then the ITS 1 if we're doing next gen. So some of our stuff is still based on Sanger because um, uh, we have problems uh, with the next gen not being very predictive of what ends up being on the roots, so that we'll sequence the roots and we'll get all sorts of other fungi, but not really some of the ectos that we know we can see on there. So I think just the bias of especially when we're going after the ascomycota. So if we were going after the basidios, we could tweak it a little bit more, the primers, but so many of our fungi are ascos. And so then you pick up all the penicillium and all the stuff, even when you clean the roots, they seem to amplify better than the ascomycete ectomycorrhizas in those next gen studies. So we often do both.
Mhm. Great, thank you.
Mhm. Okay.
We have a question in the chat from Aaron Wilson who asks if there was another data stream that you could collect in these studies, but is currently just too difficult to collect, what would it be? So it sounds like, if you could add one more metric to your experiments, like that you observe, one more thing you could know, what would you want to know?
Um, I guess what in our systems, what I would like to do is have longer grants, longer funding, and look at it longer. Because yeah, I mean, I would love to be able to look. Well, I guess there'd be a lot of things if I was not money li or time limited. So we'd obviously want to know if the plants, like for example, at Mesa Verde, will they get bigger? Will they really change the community and the soil in a way that favors establishment? Sort of that whole, I don't know if it's something that you're as familiar with in your work, but that applied nucleation, where if you can restore small areas, then you can have a bigger impact. And so that would be great to know how things change over time, over long periods of time, because we know things are slow in these systems. And then I'd also just like to be able to know mechanistically, for example, we really don't have much of an idea of why different ectomycorrhizal fungal species are good or bad. And so we know that Geopera is important for drought tolerance, and we have some studies that we were able to do at Los Alamos by putting seedlings in the particle accelerator, which is like a one-time opportunity to see that they increase water movement. But really, we don't have, we have what are the exploration types. But when you go to the Western United States, so what kind of hyphae explore the soil, that's our functional trait. But almost all of ours have sort of contact or low exploration. So it's like they got to be doing something different. And so I think having the funding to and the ability, I've tried transcriptomics without good success, but to really understand what are they doing when you're a good mutualist or a bad mutualist. And then, of course, we are exploring like, what does cheatgrass do? Why is it so awful? And why does it, I didn't show these legacy things, but just one generation of cheatgrass, if you plant pinions in there, even when it's gone, it reduces their growth by half. And changes their fungal community. So it's leaving something in the soil, and I'd love to know what that is. So that's a long answer, sorry. But
That was an awesome answer. I'd love to ask, you really leaned into bulking up these native microbial communities in all your treatments. And this is cheating because I already talked to you a little bit about this, but what do you think you would happen if you tried to just naively apply some of the existing commercially available mycorrhizal inoculants? Like if you bought mycorrhizal spores on the internet and did this experiment.
Um, well, I think it's interesting to think about it. I think I have a good prediction for arbuscular mycorrhizae because there's been so many nice studies that have shown from Liz Koziol and others who have shown that, you get pathogens, they don't work, they put fertilizer, anyway, there's lots of studies, but there's not so many studies on ectomycorrhizae. And I don't know if you have, others that you know about, but I felt like there was this time when everybody was trying to use Physolithus tinctorius in the southeast. And so there was a lot of interest, and then that waned because it wasn't the sort of be all end all mycorrhizal inoculant. And then there really hasn't been as much study. So we're trying to investigate by using one of the products that is used in the Western United States that's made out of fungal spores, but it's not any of the species that we have in our forests. And so we're basically doing greenhouse trials to try to see if it works at all, because I feel like there's not the ecto database to really know how important that is, except for studies like ours, which show that the local is better than the non-local, but we don't know exactly why. So yeah, so I think we need more ecto studies, and I don't know if any of your trials, because you're doing ecto inoculum, right, with the loblolly pine, if any of your trials include like I guess you're making custom inoculum versus buy it on Amazon inoculum. Yeah.
Yeah, we haven't worked much at all with the existing commercial inoculants that you can buy. But I've heard plenty of anecdata about them. That is very mixed. Yeah, I would agree that most of those interlaboratory comparisons are on arbuscular mycorrhizal fungi. Rather, um, call for questions, y'all. What do you got?
I have another question, kind of more genomics question, but was curious about, you mentioned, you had tried some metabolomics or like transcriptomics. Have you done any like functional gene type annotation or see any like potential changes in functional potential from inoculation?
We haven't done anything successfully yet. So yeah, our initial run at transcriptomics wasn't so good. And so we have some metagenomics data that we've just sent out, and then we're actually leaving it to others because we were trying to do some of the stuff in our own lab, and it was like, we're just not, yeah, there's too much troubleshooting to do. So we're just sending off the metabolomics, and we just got metagenomics data back. So we'll see if that helps us figure out. And that's really targeted on because we have such a cheatgrass problem here. And then we also have other bromes we're working on saguaro restoration, saguaro cacti down south, and there's a different brome there. And so we're really trying to figure out what those bromes are, what are they doing? Because they have the same effect repeatedly, regardless of the plant that we put them with, where they really just one generation, they change the soil in a way that is really inhospitable to a lot of the native plants, the dominants. So we're really trying to chase after that with those kinds of methods, but we don't have the data yet.
Hi, thank you so much for your interesting talk. I'm curious about uh, the relationship between like how far away your sort of intact forests that you used to inoculate with were from, the recipient and sort of how that could relate to the persistent of the community after inoculation.
Oh, that's an interesting question. We didn't, you could see, we just had to, because we were working at a national park for the Mesa Verde example, we had to get inoculum where they let us. And we had to get seeds that were local because that's just the park rules. And so they just said, we're putting in this so it was intact forest, but it wasn't far from a road, so it was, sort of somewhat disturbed, but we didn't have the opportunity to compare like different inoculum sources, because at Mesa Verde, that with those cultural resources, every time we took a soil sample, even just the tiniest soil sample, every time we dug a hole, we had an archaeologist right there to make sure there was no cultural resources. So it was very, so it was great. We learned a lot about, those cultural resources, but you can't just go, oh, let's go here. It's like everything has to be approved, and you have to arrange for an archaeologist to come and watch you dig every single hole. So,
Okay.
yeah. So sorry. I wish we knew more. It's just totally opportunistic. Yeah.
Totally get it. It's cool.
Yeah.
Brian, thought?
Thank you.
Brian, I think you're on mute. Yeah, I can't mute him. Brian, you're on mute. You're welcome to shoot us a message, and we're happy to pass it along later on, if that makes sense.
Here we go.
There he goes.
Oh, there you are. Great.
Thank you. Thanks to the host for unmuting me. Yeah, thanks again for the presentation. I was wondering about rhizobacteria and whether you looked at them at all in their interactions with different fungi.
No, that's a great question and a great suggestion to do that, and we haven't. We have, again, one data set from Mesa Verde that we're just putting all the primers to, so for the soil. But so yeah, that's a great question, because we're using whole soil inoculum, and maybe we're correlating how much mycorrhizae we have in those communities to everything, but it's really some bacteria that's the boss. So
Okay,
or
thanks.
bacteria, but it's a great question.
All right. Well, with that, I'd like to say thank you so much, Dr. Gehring, for your presentation and for taking some time to share with us what you've learned and what you continue to learn in restoration. And we totally got a lot out of it. So, yeah, thank you so much. And thank you, everyone, for attending.
Yeah, thank you very much for the invitation and for the conversation. I really appreciate it.